In this tutorial, we will discuss the t-SNE and UMAP analysis module. The objective of this module is to reduce the dimensions of the dataset from a principal component analysis to a two-dimensional space, using one of the two available algorithms, either the t-SNE algorithm or the UMAP algorithm. Beforehand, a project was loaded and the modules of HTO demultiplexing, quality control, gene selection and PCA were applied. In the gene selection, we performed two analyses, one with the VST method, selecting 1000 genes, and another with the mean var plot method. In the principal component analysis, we used each of these methods to perform a PCA. We see that it is possible to choose one of the two PCAs that has been previously performed. To use the t-SNE algorithm, we need to set several parameters. First, we will define the number of principal components of the PCA to be used. For this we can use the PCA scree plot which is reported here. It shows the amount of information carried by each of the principal components. The objective here is to choose enough principal components to carry a representative amount of information. For example, we can choose to stop at 10 principal components or 20. We observe that beyond that, the amount of information carried by the principal components is low. Let us choose to use 20 principal components by entering the number 20 in the corresponding field. We then need to define a perplexity value. This perplexity value is a value used by the algorithm to define the balance between identifying local and global structures. A low perplexity will indicate an interest in local structures, a high perplexity in global structures. You can therefore vary this perplexity value to observe its impact on the map created by the algorithm. If we keep the perplexity value at 30, we leave the number 30 in the field. Next, we need to give a name to this analysis. This name must be representative because you can run several analyses that can be used in successive modules. This name must be representative enough to be found. So we will call it for example t-SNE, 20pc, 30p. Then we run the algorithm. The result is a two-dimensional map in which each point is a cell. We can now use the UMAP algorithm. As before, we need to define the number of principal components to use. We will set this to 20 principal components in the same way. A second parameter to define is the number of neighbors to use. This number of neighbors represents the same thing as the perplexity for t-SNE. It represents the balance you want to represent in the relationship between large distances and small distances in the PCA space. We will leave the number of 30 and again name our result something representative. For example, UMAP, 20pc, 30n. Then we run the algorithm. We see that the result is a two-dimensional map in which each point is a cell. As you can see, the UMAP separates the cells more than the t-SNE map. The UMAP algorithm is indeed better at representing large distances between cells, which produces more separative maps. Once these analyses are done, you can move on to the cell clustering and marker gene identification module, which is the subject of another tutorial video.